I've started working on meiosis during my PhD many years ago. And this didn't happen because I planned to do so. It rather happened because I made a mutant on a kinase which would have had a phenotype in the regulation of proteolysis in cells. And what I ended up observing, and just by looking at how cells behaved, is that meiotic cells all of a sudden thought that they were undergoing mitosis. They tried to separate sister chromatids instead of homologous chromosomes. And we were not a lab that really specialized on meiosis back then, which made us have to learn so much about the process. And while learning about it from scratch, I ended up falling in love with all the intricacies that constitute meiosis. It is a beautiful sequence of tightly coordinated processes, which with each one of them being almost a field on its own. So there's really a lot to explore within the broad field of meiosis research. The different model organisms are beneficial because they all have unique properties and thus allow us to ask questions with different levels of depthness. So we use in the lab budding yeast because we can purify meiotic enzymes from their native context, which allows us to characterize them very deeply. We use mouse to explore the conservation in evolution of some of the things we have discovered in yeast, and mammalian tissue culture allows us to explore some angles related to our findings in the context of disease. I think that what is still missing is we still have very poor understanding of how DNA repair really works inside cells. We can reconstitute even some reactions in the test tube, but really simplified versions of what must be happening in cells. For example, most DNA repair enzymes, I mean, we are talking about DNA nucleases, for example, which can cleave DNA. They can't simply be left cutting DNA in an uncontrolled manner in cells. So there has to be lots of regulation, which we are still missing. We are, I would say, mostly fascinated by simply understanding how things work. It's the primary motivation is not to immediately think of ways to cure disease, but we also know that when you understand something properly to the core, it's very likely that you can come up with approaches to correct things that go wrong. And when you work in a process such as DNA repair, which is so important in preventing cancer, genomic instability, and which is also fundamental for fertility, it's of course a motivation to think that perhaps our work contributes to help people in the future. I did not want, since I was a kid, to be a researcher. It's just happened. I, like every second kid in my country, wanted to be a football player for most of my youth. I then wanted to be a guitar player, a musician. And sometime in high school, I just bumped into a very, very good set of teachers of chemistry, biology, and in particular, the lab work on the biology classes was done by a very curious PhD student who just did this as a side job to make some money, but which really made me think it could be super interesting to spend my days understanding how biological processes functions. So basic research typically aims to explore how things work without the need to generate a product at the end of the day. And as such, it is free to explore areas or topics of research for which we have no immediate application, but which may end up revolutionizing both our understanding of the world we live in as well as medical research. I mean, just look at CRISPR, for example. On a different note, I also think that basic research and teaching should go hand in hand. And teaching at all stages of university education should be done by people that actively do research, in particular basic or curiosity-driven research. I think it is important to pass on to the next generation, not just what is already written in the textbook, but also a sense of what is not known.